Afternoon and welcome you all to the third day of STAR 2025. Today's first and last lecture of the first session is on India's contributions to exploratory lunar science by Dr. Megha Bhatt from Physical Research Laboratory, Ahmedabad. To moderate this lecture, we have with us Ms. Sachana. I would like to introduce her. Ms. Sachana is a senior research fellow doing her PhD on lunar poles at the Physical Research Laboratory, Ahmedabad. Sachana has contributed to Chandrayaan-3 and Lupex's landing site work. She is university rank holder and an Inspire DST fellow. Over to Sachana.
Thank you, sir. It is my privilege to introduce our esteemed speaker, Dr. Megha Bhatt. Dr. Megha Bhatt is a faculty member in the Planetary Sciences Division at Physical Research Laboratory, Ahmedabad. Dr. Bhatt received her PhD in Natural Sciences from University of Clausthal, Germany, based on her research work at Max Planck Institute for Solar System Research. Since 2008, Dr. Bhatt has been actively involved in ISRO's planetary missions. She currently serves as the chair of the COSPA Scientific Subcommission on the Moon. Her research focuses on understanding the evolution of our solar system, particularly by understanding the surface composition of airless planetary bodies using multidisciplinary research that combines data analysis, modeling, and laboratory work. Dr. Bhatt have published over 25 peer-reviewed papers and contributed to numerous national and international conferences. Thank you, and over to you, ma'am.
Thank you, Sachana, for this nice introduction. And hello, all. So today we will start with this very interesting lecture, India's contribution to exploratory lunar science. It's very general in nature, and I'm just sharing my screen. So please confirm that you are able to see it. Is it okay, Sachana?
Still not visible at my end. Now it is visible, full screen.
Good. So the title of talk is India's contributions to exploratory lunar science. And this is going to be very, very light in content, but very interesting. And if you just ask me to summarize the whole talk, I would just, flash this particular image. Why? Because this is showing our journey to moon exploration, starting from orbiter, then attempt of landing, and then a successful landing and rover exploration through rover. So this is the image of our lander, Vikram lander, taken by our rover Pragyan. And what it shows is this ramp from where this Pragyan came down, and then a lot of experiments on lander. So this one is Chaste. Here you can see this seismometer and some cameras and all. From this particular picture itself, there's a lot of things, very interesting facts, what you can learn about moon. For example, the terrain, the type of terrain, the kind of differences in the morphology or the surface structure, you can say, and that gives us perspective. So we would not get this kind of understanding if we would have not started with exploration. So that is the importance of exploration. And obviously moon is the nearest to us. So moon serves as a natural laboratory for us because it's a bit easier to reach there compared to Mars or to Venus, which are also equally interesting objects. But moon is kind of, natural laboratory where you can go, you can experiment, and you can learn for further exploring our solar system. So let's start with moon, because obviously I don't have to just conclude in one slide. So I go ahead with this. So if you have seen moon, and if not, today is the best time because today is full moon, and you should obviously look outside tonight, and you should start observing by your own, because your eyes are also doing remote sensing. So basically, if you look to moon, you are familiar with this side of the moon. But this side of the moon was unknown to us till the first spacecraft, Luna 3, provided us the images of the far side of the moon. And the interesting fact is that both these sides look quite different. Until we did not have this image of the moon far side, our understanding was that the far side or the back side of the moon should also look very similar to its near side, or to the side which is facing to Earth. Because there is no, why we should expect these kind of differences? And it is puzzling that we do see this kind of differences. So the first time when scientists started getting these images, we were wondering, is it the correct image we are looking on? So this is quite interesting about moon, and though it is so near to us, but there are still many unknowns. But along with unknowns, because we have a systematic program of exploration, and we started in 2008, but before that, from NASA and from USSR, we had series of missions, and those missions also the telescopic observations provided us wealth of data and a basic understanding. So based on that basic understanding, we know that the moon spins on its axis and rotates almost at the same time, once in every 27 days. And that is the reason why you only see one side of the moon, not the other side of the moon. Okay. So this synchronization means you must have heard that Chandrayaan 3 mission was planned for one lunar day, and that one lunar day means 14 Earth day. So that means that at any particular time, if you are on moon, you will be having 14 consecutive days of sunlight and 14 consecutive days of night. Okay. And then another interesting fact is, we know now that moon is drifting away from Earth. So does it mean that moon was very nearby to us in the very initial phase of the evolution of Earth moon system? This is again a very interesting question. And because we have, even Chandrayaan 3 had a seismometer on board, and then other missions, also Apollo missions, had a seismometer, and we know that moon also has quakes. Though moon is one plate planet, but it also has shaking. So the concept what we had initially, that moon is kind of, nothing is going on the moon, is not true. Now we know with our exploration plans that that is not the correct statement. So moon is also shaking. Moon is also basically, lot of things are going on the moon, which we need to understand. Another very interesting fact about moon is water on the moon. All of you must have heard about it, but no liquid water. Okay, so it is not in form of liquid water because there is a perfect vacuum on moon. So you cannot have liquid water on the surface, but you can have water in the form of ice and mixed with soil or regolith. And this understanding is also very recent, because initially our understanding was that most likely moon is bone dry. So there is no water, but the new series of experiments, exploration provided us detail that no, that is not the case. We have to revive our understanding, and we have to, come up with next set of questions that there is water on the moon, and if that is the case, how we can utilize it. Because if we are planning for base on the moon, or if you want to have resources from the moon, water is something which you would like to extract and use. Another important fact is that initially it was thought that moon is a airless planetary body, and that is still we are continuing with that thinking, but we have enhanced on that in terms of understanding that it do have a very thin and weak atmosphere. Okay, and in this particular direction also Chandrayaan missions have contributed significantly. And then the surface of the moon is fully covered by a very fine layer of dust, or in scientific term we say regolith. It's very similar to what you see in case of Earth soil. And the main surface alteration process, for example, on Earth is rain, wind, and so on. But here in case of moon is space weathering. And if you understand space weathering on moon, basically this information or this knowledge you can extrapolate to understand any airless planetary body like asteroids. And then we have a very good understanding on, but not yet fully understood, is the fact that moon must have formed from a giant impact. So a body might have impacted on Earth, a very huge, like Mars size body, and that might have actually formed our moon. So there are obviously a wealth of information, and this new data set, new information has opened up new questions. One more interesting fact is that if you look from here, the face of moon would look something like this, okay? But if you are somewhere in the other part of the Earth, you might see this particular crater, what you can see here on top, okay? So for example, if you are in a new place, and well, now these are not the conditions because you are very much, aware with internet and all. But if that happens, that you want to make yourself familiar with orientation, how would you do? Actually, moon is something which is visible from everywhere, right? So, but if you look from, other side of the world, you may find that the face looks still the same. So the moon is still showing the same face of the same face near side of the moon, but the orientation is upside down. So this is from, let's say, from India, but maybe when you move to other side, and you may see it upside down. So here there's a case. So this is from northern hemisphere, and this is how you would look full moon from southern hemisphere. And what would be the case if you look from equatorial area, exactly, equatorial of the of Earth? That is a question which you should be coming up with the answer. So why at all we should be exploring? So one question is that now we have our systematic exploration path, but why should we explore at all? So here I would like to quote Bill Andrews from Apollo 8 mission, and I would just read this because it's very beautiful quotation, what he summarized very nicely. We came all this way to explore the moon, and the most important thing is that we discovered the Earth. So whatever exploration is there, it's actually related to our own world, because we want to protect, we want to understand our world. And we are curious to know for other habitats. So we would definitely like to extend it for searching for other habitats. And planetary exploration helps to develop new technologies, and that further can be, used for economic prosperity. Obviously, we are curious all the time, and we would like to know answers of unknown questions. And with this kind of missions or exploration plans, we team up with international people, and that enhances international cooperation, trust, exchange of culture, and lot of other things which we learn by sharing with each other. So this is why it is important to explore moon. And then, as I discussed already in previous slide, we are curious. So now I'm moving from, I'm trying to take you towards scientific, objectives of exploration. So in case, particularly in case of moon, I have listed down these 5 top questions which comes in my mind. Obviously, there can be plenty of other questions, but if I have to categorize, I would say that we still have to understand about origin and evolution of the moon, because when we talk about moon, it is basically the Earth-Moon system. So because moon is affecting Earth, and you know that we are getting these tides on Earth, and basically could be that we are having habitability, or we are having life on Earth because of this tidal effect, which is like, heavily mixing, churning, and providing, exchange of energy. So in that case also, moon is a very important object to understand, because when we talk about moon, it is basically the Earth-Moon system which we are trying to understand. So origin and evolution of the moon, and if I further divide it, I would say that we would like to know how did the moon form. We have some idea that this is most likely due to giant impact hypothesis. So basically a Mars size object hit Earth, and there's mixing of this, and this particular such a huge event uh, generate a disk of debris, and that eventually at a certain distance start forming the moon. But there are many uncertainties here, and there are several models which tells that could be that moon is made of mainly the mantle of Earth, or maybe the maximum contribution from Theia, and because we have some kind of similarities between Earth and moon, but there are certain dissimilarities also which we need to still understand. So this is something which we still have to find out. Then we also have to understand that is it that early Earth, uh, I mean basically what kind of information we can get from moon about early Earth, because Earth is an active planet. So here we do not have all those evidences. But once moon is found, and because moon is not very active, that's why whatever information is stored there, it will be staying there as it is until unless you disturb it. So that's why moon is like a kind of place where you can try to get the evidences, the old evidences. And is there, like, when this kind of kartic formation happens, would there be a fully molten magma from where the crystallization starts, or would it be a partial melting? Because that has implication towards understanding its evolution and its geology. So these are again, part of questions which we need to understand from origin and evolution of the Earth-Moon system, I would say. Then lunar volatiles. So obviously, when we talk about volatiles, the first thing comes in mind is water. But water is not the only volatile we are looking for. There are several other volatiles which are very interesting to explore. For example, there is sodium, which is like, you can basically, now we have from Chandrayaan-2 mission, we have sodium maps for and it at the global scale and so on. So there are several other volatiles which are very important to understand, and these volatiles are mainly important from understanding the accessibility of these resources. So how accessible are they? What is the origin of these volatiles? So if you think that there would be, like, too much of heat during this kind of formation, chaotic situation, would there be volatiles still remained in the very initial time? So is it like contribution from at a later stage, or is it from the time of the formation? So origin is also not yet fully understood. The distribution is not yet fully understood. And how can we map it? So that is also something which those Chandrayaan missions have significantly contributed in that direction, but there is a lot which we still have to understand. Then permanently shadowed regions, you must have heard this particular term. These are the areas which are always in shadow because of because moon is almost, there is no tilt on its axis. So because of that geometry, there are several areas which are always in dark, like this particular example. So when you have this kind of area sitting in shadow for billions of years, there's a high possibility that you may accumulate volatiles there. So that's why permanently shadowed regions are very important to explore. And this is a reflectance image of this particular crater, Shackleton crater from the southern lunar pole, which shows that what kind of morphology do you expect in this kind of permanently shadowed region. So do you expect eyes there or not? The results indicate that yes, but then there are several results, for example, even this one, where we do not see any difference in morphology, which can tell you that there is water ice present. So it is still a very much open question, which we have to understand. Then if there are volatiles, what kind of transportation mechanism, or what kind of accumulation, how do you get it and how do you basically release it? So we have some idea from modeling and from observations, like we know that the solar wind from sun is basically contributing towards getting this superficial water ice that eventually, if it goes and reside in these kind of places, it stays there undisturbed. But then there are several hypothetical elements in this particular graph, which we still have to understand. So basically the overall cycle, though we have a good understanding now, but still there is a lot which we have to understand. Then lunar interior. We have some idea from samples because we basically extrapolate it towards the interior, but there are still many open questions. For example, what is exactly the composition of mantle? What is basically the boundary for mantle and crust? And is there molten core in case of moon or not? So there are several questions in that direction. Geological history of the moon. So for example, as shown here, initially might be that there were impacts like this, and then you got some kind of volcanism on the moon, and after this volcanism again some kind of impacting. So this kind of modification processes are there, which are in a chronological order, very important to understand the overall geological history of the moon. And when we say moon, obviously we are also trying to understand Earth-Moon system in an integrated way or in general way. So in a very, very like, broad terms, moon exploration is very interesting, because moon is, you must have seen that now the interest is again revived and everybody is behind moon, because moon, initially we thought that okay, we have learned a lot about moon, but then a new set of questions uh, indicated that there's still a lot which we can do and we should be doing, because again, because this is a nearby object for us. So there is a lot right now going on in terms of we might use moon for mining resources, for example helium-3, which can be used for getting the nuclear energy sources and so on. We can use moon as base for further exploration. If we use moon as base, obviously what we need is to use moon's resources for fueling, and then we should have a permanent settlement on moon, because if we want to further move out in solar system, moon is the first challenge. So if we can have a permanent settlement on moon, we can think about going beyond moon, and also orbiting space base, and then space tourism. These are the new interesting branches now coming up with lot of excitement. So here in this particular slide, I want to summarize that moon exploration. For example, here what you see is at the near side, these are the landing sites listed here, and Chandrayaan-3 has landed in the very much southern latitudes, southern high latitudes, highland latitudes, and these are the upcoming moon missions from governmental organizations and private organizations. And as you can see that we also have 2 missions planned, Chandrayaan-4 and LUPEX Chandrayaan-5. The main long-term goal for all these exploration plans are basically to have a permanent base on the moon. This can be, an international effort also, and in order to have these natural sources, water and oxygen, precious metals, rocks and rare earth elements, and helium-3. So these are mainly the the long-term plans. And as I also already said, that the moon poles are also very interesting. For example, here the south pole of the moon is shown, where there are several, the shadowed or the dark regions are the permanently shadowed regions, which could be obvious shield for us, and these are the areas where you can have the highest probability of having water ice, and that can be used as a resource. Now coming to our own systematic plan or our own contribution, we have total 3 missions so far, and 2 more are planned, as I already said. And we started with Chandrayaan-1 in 2008, obviously planning and the overall framing the mission and all started much before than 2008, maybe in 1990 and so on. And the first mission was a truly international collaboration, where we had several instruments from India, whereas a few, for example, the SKICS and SARA in collaboration, and then a few like SIR-2, M-Cube, and MINISAR from outside India. So this was a truly international mission with the total 11 payloads. So you can see that actually, if you look to the name of these payloads, you will understand more or less the objective of these these instruments or payloads. For example, terrain mapping camera to have very high resolution images and to prepare a terrain map of the moon, then hyperspectral imager for looking, at a very high resolution, the surface of the moon, the surface composition, the mineralogy of the moon, and so on. Also, this KICS, or this Chandrayaan-1 X-ray spectrometer, working in X-ray regime for compositional understanding, elemental understanding, and so on. So a very nice package of instrumentation. So why do we go with so many of instruments at one go, and why you always look for different wavelength ranges? So this is again a question which should be asked and which should be understood, because you will see the same pattern in Chandrayaan-2 also. So here also we have a high resolution camera. This one was specifically for looking from the oblique views and mainly for mapping the landing site. Then we had the advanced version of terrain mapping camera, advanced version of KICS here, and this IRS advanced version of M-Cube, and so on. So we are always doing multi-wavelength measurements. So basically what we are trying to do is we want to look to moon in different wavelengths. Why? Why it is so? So that is again a question which you should be thinking about. And then Chandrayaan-3 in 2023, where we had a set of instruments on lander, this seismic activity instrument ILSA, and then Rambha, Chaste, and then we had 2 experiments on rover, APXS and LIPS for elemental and mineral mapping, and then we had this shape on the propulsion module to understand the the signature of a habitable planet. So looking to Earth from the moon orbit, considering that if you are looking to an exoplanet, what kind of signature you should be expecting. So here is the answer to the question I just asked in the previous slide, that when you look moon in different wavelengths, you get different information. Okay, so starting from here, from the longer wavelengths, basically, starting from radio and going to NIR and then to visible, UV, X-ray, gamma, what you see is that you are getting different kind of information for every region of the wavelength. So when you integrate this information, you get the overall information or in-depth information of a specific site or a specific body. And that is the reason that we always go with multi-instrument, looking at different, some are looking to maybe atmospheric composition, some are looking to surface, some have the capability of understanding the inner structure of a planetary body, and so on. So these are the instruments very meticulously designed and selected, and you have lectures on these aspects, so I'm not covering that in very much detail. So now I'm basically focusing on the scientific part. So what is the contribution of Chandrayaan missions to new views of the moon? So there's a significant contribution, and I want to start with the one specific case of technological innovation. Okay, so obviously we are more interested towards science, but science and technology go hands in hand. So basically, we are improving in both the sides with every subsequent mission. So every subsequent mission, we are developing new technology because we are given new challenges. So new technology, new understanding, and based on that new understanding, new scientific questions. So here is an example from Chandrayaan-3. If you look, that it had basically I would say that this is a technological innovation where we had multiple backups for Chandrayaan-3. successful landing, and it was fully automatic landing. We did hop experiment, and that demonstrated ability to take off from a celestial object and land again. Another technological innovation was bringing back the propulsion module of Chandrayaan-3 from lunar orbit into Earth orbit. This was also successfully done, and testing technology for future interplanetary missions, because we wanted to use this opportunity for upcoming missions. And these experiments have taught us a lot, and the data what we have collected is basically a base for further improvement and for innovation. So here is the image. This one is again, both of them are from lander camera, and what you can see is the difference. So these two craters, if you focus in this particular image, you see them shifted, and this is a reference. So this particular reference is there in both the images. So what you see is this is, and these are the rover tracks, and what you see is that this image is before hopping, and this one is after hopping. And with these images, we confirm that this hopping experiment was done successfully. So where are we now? So we, for summarizing this, we had a first successful landing in 1966. We had a human landing in 1969. We have now return samples from Apollo and Chang missions, and then we have a first successful Indian landing at the high latitude region. So this is again first, which is credit to us. Now coming to a few scientific results, and I will be going a bit fast now because I have just 10 minutes left in my hand. So basically, the most important result from Chandrayaan-1 mission, and that is followed by Chandrayaan-2, is confirmation of water on the moon, discovery of water on the moon. So there were indications from previous missions, but this was the first time from Chandrayaan-1 where we saw that as you are moving towards pole, you have this, this is from the hyperspectral imager, and that indicate the presence of water. And this was also confirmed by looking to MINISAR experiment again on board Chandrayaan-1, where we looked to the CPR ratio, the circular polarization ratio, which is a parameter to look for the water ice presence, and there were a few anomalous craters which showed enhanced CPR signature, and that basically indicate that there is a high possibility of having water ice on the lunar poles. Another direct evidence came from our own experiment, Moon Impact Probe, which had a instrument chase on board, and that provided the altitudinal variation. So for example, here hydrogen, this is argon. And what you can see is that as you are moving towards pole, the density increases. So that again indicates the presence of water in form of ice, and these observations were also correlated to M-cube or to the hyperspectral data, and it was found in good match. From MINISAR, we have PSR regions which you cannot simply photograph. Those are the regions which you can look from the radar imaging. And this is such one case where you can see that, there is a decomposition of the scattering parameters, volume scattering and backscattering and so on, where you can see that, different colors basically tells you a different kind of scattering phenomena, and that indicate you the soil properties. And here this is the CPR map, and this red areas basically indicate a possibility of having water ice. So from this particular instrument, we could have the catalog of several such craters which could not be examined by the optical cameras. And this more than 95% of polar region was basically mapped in a systematic manner. We also have supportive evidences. So here, for example, a study where not only enhancement in the signature is reported, but also the variability based on diurnal variability is also shown, where in morning you have more water which evaporates, and then by evening you start getting that back. And these are the polar areas where again map is prepared using by integrating data from different sources, which shows you basically the potential sites where you may look for water ice exposures based on these kind of signatures. A new rock type was also discovered using Chandrayaan-1 moon mineralogy mapper spectrometer, and this was quite surprising because this was not actually found from the Apollo samples, and this was basically first reported from this particular area of the moon. And later on there were systematic studies which also reported this kind of lithology from other different areas, and that this particular mineral, pink spinel or the Mg spinel, basically is the one which gets crystallized at the early stage of crystallization. And because of that exposure, one can try to understand the early history of the moon, and this has basically implication for understanding the evolution of the moon. And that how do we get these kind of information is by looking to this kind of spectral signature from this reflectance versus wavelength graphs. Another very interesting study or discovery class result is from, again from Chandrayaan mission, is this young volcanism and extended volcanism. So initially it was thought that volcanism stopped something like 3 billion years ago, but the new information, high spatial and spectral mapping, shows that that is not the case, and you may actually get this kind of features which explain that moon is basically active of something so young as something like 50 million years. So a lot of systematic study has been carried out using Chandrayaan datasets. Another catalog prepared for domes. So these are again volcanic features, and for looking to them, you have to look to its morphology, and for that, what you need is the elevation map. So this kind of slope map or this kind of 3D maps are prepared to understand the parameters of domes, and several of these domes are basically reported using Chandrayaan data, and a catalog is prepared. The spectral classification is used for looking to the elemental composition, or a very detailed spectral classifications carried out using again Chandrayaan data. And high resolution maps are prepared now, where we have information of distribution of iron, calcium, magnesium, and a few other elements, which basically tells us about the surface mineralogy and the surface composition, that again can help us to understand the interior of the moon and how this might have evolved. The lunar environment initially was thought very, very, simple, but now after Chandrayaan mission, we know that it is very complex, and a lot of dynamics going on from the day side, night side, and interaction between day and night side. Another very interesting result came out from Chandrayaan mission is the discovery of mini magnetosphere, the strength of the mini magnetosphere. And if you have this kind of mini magnetosphere, because moon do not have the full magnetic structure like Earth have, So why do you have these remnant magnetic fields, and why do you have it, and what is the origin, and basically what is the relation with the surface feature? So these are again something very interesting findings out of Chandrayaan mission. Now, specifically looking to Chandrayaan-2, which had orbiter, lander, and rover. I will very quickly go through it because I think, yes, I had just have 5 more minutes. So this was the picture we had before Chandrayaan-2, where a very sparse coverage in X-ray, which basically is a direct way of getting elemental composition. But because of the limitation of this technique, we had a very limited coverage from X-ray spectrometers, but Chandrayaan-2 provided us the first global coverage, and this particular coverage is now substantially revised, and we have even better coverage now from Chandrayaan-2. Global elemental maps are prepared: magnesium, silicon, aluminium, iron, and also sodium maps are available. So this kind of new information have been retrieved from Chandrayaan-2 mission. This kind of detailed landing site studies have been carried out using Chandrayaan data set, Chandrayaan-1 and Chandrayaan-2. So here you can see the alternate and primary landing site of Chandrayaan-3, and what kind of composition and mineralogy you expect, or what kind of thermal behavior you may expect. So these kind of detailed analysis are required before you send your before you land. So these are also, carried out using the inputs from our previous missions. So I will just skip this particular thing, and then IRS basically from Chandrayaan-2 has provided information on OH H2O mapping. So 2 specific studies have been carried out for which tells you spectral unmixing of this, basically telling you the type of minerals you have, and then the complete characterization of this hydration feature in and around the Shiv Shakti point. And this kind of data sets are very complicated, so you need to spend some time to understand these kind of data set and basically to reduce them in order to get the information, extract the information. I will not go in detail to these things because I already explained from Chandrayaan-1 point of view. Now with Chandrayaan-2, we have dual frequency SAR, which gives you even further information because it gives you all the parameters which help you to basically bifurcate the information and prepare precise CPR maps and basically, the decomposition matrix that can be related to a specific kind of scattering phenomena. So this kind of, detailed maps are prepared. Now coming to Chandrayaan-3, next level of complicated mission, and this was basically the one lunar day measurement, and it provided us a very wealth of information from because it's an in situ measurement, you can say. We have now a good understanding on the thermal profile of first a few centimeters, and this ILSA basically help us to understand that.
Ma'am, you have 2 minutes left.
Okay, fine. So some movements which are like related to known activity and unknown activities and so on. So very like very impressive landscape. I will just skip this. Another very important result is from APXS on board Chandrayaan-3, which basically provided us a systematic mapping of 100 meters, and it provided us the information that overall the composition is not changing within 100 meters of the track traveled by the rover, and this is a typical spectrum shown here, and the chemical composition found out of this. So presence of several major and minor elements, and now this particular instrument information can be used as a ground truth for other missions. So I will simply skip this, and now basically now what is the direction? So basically we have some understanding on sources of this, volatiles, and how do we basically remove this volatiles. So basically sources and sink processes. But to further understand it in detail, you need landing missions, or you need to go to the extreme south, and in that particular direction now the Chandrayaan 5 LUPEX mission is planned, where we did a systematic study and look for the landing site where we can basically land, and we can get maximum information about these kind of volatile cycle. So I will just leave you with this particular image, where this shows the initial time of exploration. So this is the near side, far side of the moon, the first far side image, and the recent image, and you can see the difference. Though this is also equally important, but you can see the finer scales detail what we can now get with the much better instrumentation and much better understanding what we have developed from 1960s to now. Thank you, Sachna.
Thank you, ma'am, for keeping it on time. We can take 2 or 3 questions due to time constraints.
Yeah.
So, yeah, Chandni is asking, are the samples received from the moon in lunar mission aligned with the age of mountains or rocks of the Earth?
Age of, okay, the thing is what we get age is actually quite before, because here on Earth everything is very much dynamic, so it's very difficult to get the very old ages. But whereas with Apollo samples and lunar sample, we have a very good idea about that when these features are formed. So basically, obviously you see a synergy between these two, but you get much more information from these Apollo samples about, like, what would be the conditions in at a very early time of evolution of this Earth Moon system.
And also there is one more question from her. So moon is moving farther from Earth day by day. Does it cause any disbalance in the solar system stability?
Solar system stability, well, I would say that we have to first understand our Earth Moon stability. So that is quite important to understand. And in this particular direction, again, there are two school of thoughts, because there some analysis show that, okay, moon is important to keep our Earth on its axis, and this tidal effects we are getting because of moon. Other school of thought is that, okay, moon is important, but is not the important to have habitability on Earth. So this is again a open question, which is a very important question you asked, Chandni, right? You said so. Yeah, whatever. So sorry, I am bad with, like, pronunciation, name, and remembering name. But this is again a topic where a lot of research is going on. Chandani, yes.
So Urvi is asking, what is the mechanism responsible for the formation of water in the form of ice if moon surface is vacuum?
The thing is, comets are bringing a lot of volatiles with it. So first thing is, if these volatiles are somehow reaching to poles, it will stay there for a longer period of time, because it is like a permanent fridge there. And then also, as I also mentioned, that this could be from the interior also. So initially our understanding was that this moon was a ball of lava, and there was no way that you can have volatiles during this process. But the new models suggest that there is a possibility to have some amount of water which you can get. So that is basically the what is called endogenic water. So there are endogenic as well as exogenic sources, and it is very important to understand both of them. On top of that, solar wind is also contributing, because solar wind is a major source of having hydrogen and oxygen and so on. So basically this oxygen, or which is on the surface, when it reacts with solar wind, or basically the oxygen associated with the other elements, when it reacts with solar wind, it can produce water, which eventually if migrate from equatorial to polar areas, because there is a thermal instability, in the sense that you have equatorial at very high temperature, whereas the polar region will be low in temperature. So obviously these molecules will move towards pole, and if they reach to permanently shadowed region, they will stay there for a longer period of time.
There is a follow-up question for that. How we detect the ice if it is in the ground?
So how we detect ice is through several different methods are there to see this. For example, here in this particular talk, I talked about MIP experiment, this chess outcomes. So here you directly look it in terms of AMU measurements, where you can basically find out that okay, you have a signature for hydrogen and for argon and so on. Then you can look through near infrared spectroscopy, where you know that if water is present, you will have some specific signatures. So you basically try to look for these signatures, and you try to get the information of the depth, and that can basically tell you about the abundance. And then you have this DFSR, or microwave instruments, which basically again give you information about the presence of water ice, because like we discussed, the CPR is a parameter which tells you if the value is high, that basically most likely because of the presence of water. So these are different techniques that we can use to detect water ice on them.
One last question. Why there is an absence of atmosphere on moon?
Because moon do not have its shield, so it doesn't have its magnetosphere. So solar wind directly, earth is having this shielding, so that this moon sun solar wind, or the sun high energy particles are basically not directly interacting to us. So this atmosphere is there because of this outgassing. So in case of moon, because the shielding is not there, it is lost in space. So that's why you do not see this. And then the gravitational force, so it doesn't have that high gravitational force, it's 1/6 of Earth, that it can keep these gases associated to its surface. So that is the reason why you do not have atmosphere.
Thank you, ma'am.
Thank you, Sachina, for nicely moderating the session. And also our sincere thanks to Dr. Megha Bhatt for a well-educating and informative talk on India's exploratory lunar science program. Thank you once again. Now we are going to hear on the 5th lecture and the first one in the second session on steps towards Space Vision 2047. And there are 9 lectures in this session. The first lecture in this session, that is on microgravity and biology of living systems, challenges for human space exploration and new opportunities for research. This is the first lecture under this session. It will be delivered by Professor K.G. Sreejalekshmi from Indian Institute of Space Science and Technology, Tirunelveli. And I would like to introduce Ms. Chitra R. Nair, who is going to introduce the speaker and moderate the session. Ms. Chitra R. Nair completed post-graduation in chemistry from Mahatma Gandhi University, Kottayam, Kerala, currently pursuing PhD at the Indian Institute of Space Science and Technology, Tirunelveli, under the guidance of Dr. K.G. Sreejalekshmi, the speaker of today's lecture. And the areas of interest of Chitra R. Nair are organic synthesis, entrimer chemistry, and thermognostics. May I invite Ms. Chitra to introduce the speaker and moderate the session. Over to Chitra.
Thank you, sir. Good evening, everyone. It's my privilege to introduce the speaker, Dr. K.G. Sreejalekshmi, currently a professor in the Department of Chemistry, Indian Institute of Space Science and Technology, IIST. As one among the founding faculty members of IIST, joining the institute on its very first day, got the unique privilege of being mentored and guided by the luminaries and visionaries of Indian space program, including Dr. Abdul Kalam, Dr. G. Madhavan Nair, Dr. B.N. Suresh, to mention a few. Her thrust area of research is molecular engineering, which is put into action for the combinatorial design and synthesis of smart and functional materials for space applications, drug discovery, and development. Very recently, her group also initiated research in the areas of gravitational biology and space life sciences. Currently, her research group is developing microgravity biology payload to fly in the developmental flight of Indian human space mission Gaganyaan and another biology payload for the joint ISRO-NASA mission to the International Space Station. She also mentors a couple of startup companies in the IIST, including the Spacetime 4D printing company, which offers 3D printers and printing solutions. She is also the chief coordinator of the Space Technology Innovation and Incubation Center STIC at IIST. She is a member of several national and international professional bodies such as ACS, ISNM, MRSI, IEEE, SPSI, and so on. And also served as a member of committee for Vision Plan for Microgravity Science for ISRO, and also is a member of HRDG Microgravity Subgroup ISRO. With these words of introduction, let me invite Dr. K.G. Sreejalekshmi.
Yeah. So thank you, Chitra, for that very brief and concise introduction. So I will start my presentation. Before that, I would like to share my slides. Hope my slide is visible now.
Yes, ma'am.
Okay. So yeah. So let us start the presentation, and I would request Chitra to just prompt me when 30 minutes of my lecture is over, so that we can wind it up on time. So to all the participants, namaskar and good afternoon from Indian Institute of Space Science and Technology. So let me start this session by thanking the organizing team of START 2025 for giving this splendid opportunity to talk to you in this auspicious program. So considering the imminent missions and programs of ISRO, which will involve taking life into space, which is for the first time our country is doing, I have chosen that particular topic. As you can see over here, I'll be talking about microgravity and biology of living systems. So specifically, the talk will touch upon the challenges of human space exploration and the vast ocean of opportunities of research, education, and of course, giving back to the society while supporting the country in its space program. Let's proceed with the next slide. Yeah, so before going into the crux of the presentation, I would like to share 2 or 3 sentences about my institute. So we represent the Indian Institute of Space Science and Technology. Many of you must be knowing the institute. It is very small, only 17 years of age, and we are having a deemed to be university status, and we are under the Department of Space. And we have a unique mandate of undertaking teaching and research in areas that directly impact and support the space endeavors, especially for our country. So in that context, we are an academic institute as well, offering BTech, MTech, PhD, and postdoctoral programs in areas that are closely related to space technology, space science, and space applications. So moving further, now this is about today's lecture. So at the outset, let me tell you that the contents of this particular lecture has been kept as simple as possible so that every passionate space enthusiast and the budding space researcher who are attending this session will be able to travel along with me. Okay, so in the lecture, we'll be starting with the unique features of a space laboratory and the challenges when it comes to the human space flights. Okay, so then there are areas which the country needs to build capacity upon. Those are the space biology and the bioastronautics domains. I will talk about what these two domains are telling you about. And then to prepare ourselves to meet the future requirements of our space programs, we have to be ready with some of the knowledge related to the microgravity research and the outcomes and why and how we can perform these experiments. So with introducing to you some of the microgravity research platforms available within the country, I will also touch upon the prospects and opportunities for learning and research in these upcoming domains in our country. So the first question is a very, very unique question, and many of us have already asked, or maybe we are trying to answer the particular question, why we go to space. Yes. So the thing is, we already know that space is a very harsh guy, right? Despite this harshness, we want to go to space. So what is the reason for that? The simple basic reason is to unveil the secrets of the universe. We want to know what is in there. So in that pursuit of understanding our pursuit of understanding the secrets, we actually are expanding our knowledge. Okay, so once we try to break certain boundaries and go beyond, we are trying to expand our knowledge and in that journey, we knowingly or unknowingly inspire a lot of the next generation of academicians, even artists, engineers, scholars, scientists, thinkers, whoever coming in the way, we are able to inspire them. And during this process of exploring the space, we also make groundbreaking discoveries and which help us to bring the benefits back to humanity, back to society. And in that route, we also create a lot of new opportunities. So by now, we should be now convinced enough that why we go to space is actually a thing that we want to do and we should do so that we can actually contribute back to the society in all possible ways. So this is one slide. I think everyone in this particular forum will be actually mostly proud and happy when we hear this term, Gaganyaan. So it happens that we are the lucky among the citizens of the country who are living this time to see this great missions happening from Indian soil. So Gaganyaan happens to be the India's first human space mission, where the project envisages the demonstration of the capability to undertake human space flight by the country's space research organization, Indian Space Research Organization, or ISRO, what we call it as. And we are demonstrating our ability to launch Indian astronauts from Indian soil on Indian launch vehicles, and then taking them to a low Earth orbit, what we call as LEO, of around 400 kilometers away, and on a 3 days mission. And once the mission is complete, bringing them back safely to Earth and landing them in Indian sea waters. So everywhere it is Indian, Indian, Indian. That means that the technologies, the readiness, everything has to be well handled within the country. Of course, we are learning from many others, but in the long run, we have to develop capabilities. So when we talk about capabilities, we are really proud that ISRO has made every Indian citizen proud by establishing or by building its capabilities in several sectors, including space transportation, satellite systems, or space applications, and so on. But when it comes to this human space missions, we are now evolving new domains, what we call as human-centric domains. So current focus of ISRO, a large group of its scientists, engineers, is to develop expertise and capability on this, what we call as human-centric domains, where the whole organization and all the partners are trying to build upon human-rated platforms, including human-rated launch vehicles, then life support systems to support crew on orbit or crew in the space laboratories. And for that, training facilities like astronaut training facilities, who has already established under HSFC, we have an astronaut training and research facility in Bangalore. So these are all the domains which are further looked upon by ISRO for people to take up the challenges and support in its missions. So apart from that, in missions like Gaganyaan, where human subjects are involved, there will be a lot of missions that are preceding. For example, at least 2 developmental flights. Actually, it is now more in numbers. So we will conduct experimental flights to assess the performance of the flight, so that whether any alterations or whether any changes need to be built into the system to bring the humans safely back onto Earth. That is the primary objective. So along with these missions, what we, the scientists or the academicians or the researchers get is a host of opportunities to conduct space science and space technology development experiments. So I'll be talking about some of the opportunities that are coming up as and when we proceed with the presentation. So before that, we already know that our space is a harsh guide, right? So definitely life into space is going to face a lot of challenges. So there are conditions like extreme physical and environmental conditions, radiation conditions, microgravity conditions of space flight. So for example, lower gravity levels, what we call as the microgravity, in space as well as on celestial bodies, we'll be knowing that Moon is having one sixth of the gravitational force, that other celestial bodies are having less and less. So typically we will have all these impacts will be brought upon like harmful impacts on humans and as well as other living organisms that are taken to space. So to precisely tell a few of them, the variation in acceleration forces when the launch is happening, when it is staying in orbit and coming back, as well as the confinement when it comes to long duration missions and isolation in similar cases, the microgravity conditions in orbit and the radiation exposure, all these are actually very, very challenging conditions for the life which we take from Earth to space. So they all take their tolls when this human space or the life into space experiments are happening. And apart from that, when we are in space, we are away from our own mama, right? Mama Earth. Our Earth is, we are away from Earth. So it happens that we are no more having food, water, or even air. And unless otherwise you carry them in sufficient quantities, you will not be able to survive in space. And carrying them and replenishing these resources are again a very costly affair. So we have to have regenerative, sustainable systems in space. That is yet another big challenge that the country is going to face in the coming days. So when it comes to human body, initially we face a lot of challenges, small effects like space motion sickness is something, then headaches and congestion due to this shift in the body fluids because of the lack of gravity, as well as lower back pain and the adaptation problems with microgravity. So we face a lot of challenges when life is taken into space. So if we know that we are going to space and we need to keep ourselves healthy in space, understanding the challenges is the first step. Okay. So as I told, many of the challenges are already listed. So here the 5 things that is we are seeing in box on your left hand side, which is marked A. It talks about 5 very specific health hazards in space, which is listed by NASA. It includes space radiation. Then, of course, altered gravity effects, the closed environments, which are mostly hostile, and of course, the distance from the Earth, which actually causes a lot of troubles when it comes to the healthcare for our crew members, as well as delays in communication, as well as the isolation and confinement, which will actually make a lot of impact on your circadian rhythms, your behavioral impacts, your team dynamics. All these things are very, very important risks or hazards that we have to consider. Apart from that, when you look into the right-hand box where I have listed few so-called individual factors. So we know that we have a list of risks associated, and apart from this risk which is already there, we are introducing individuals into these human missions where they have their own lifestyle, their own microbiome, their own genetics and epigenetics. The age will be different, the sex will be different. So on combining these 2 things, what we call as the human system risks and the individual factors contributing to these particular challenges, we have a term called space exposome. So typically it is a unifying framework which talks about the interaction of all the environmental impacts on the human body combined with the individual factors. So exclusively the space exposome is going to define whether the human system or the space travel is going to be a successful one. So these are things which are individual centric as well, along with all the available changes or all the available environment in the space which are to be faced by the human body. Then comes one more slide which talks about the biological features of space flight. So essentially, the right-hand side box talks about the space risks that we have covered, including distance, confinement, the hostile environment, gravity conditions, and radiation. And you will be finding some of the links to certain articles, research articles, which will give you further more details about these particular experiments in detail. This is actually conducted by NASA. And on the left-hand side, what is shown as two sets of experiments, because most of the time we will be conducting analogous experiments on Earth in order to find how certain organisms or certain bodies will respond in space. So on the green shaded portion, what you see is what you call as the Earth analog experiments, where we will be using, say, humans, or maybe plants or non-human animals or even cell cultures to understand the impact of, say, radiation or microgravity or confinement conditions on Earth. Okay, so similarly, you will have also experiments conducted on so-called space laboratories. I'll come to some of the space laboratories available within the globe at this point of time and what is going to come up. So on the left-hand side, this radiation or this space-related research also takes the similar subjects for the studies. Okay, so once we get the data on these two sets of experiments, you can actually analyze the result. And with respect to the sample or the subject on Earth, how come the in space sample is different? So collating that information and data, you will be able to bring up or build certain, the fundamental knowledge related to space flight. And using this knowledge, both fundamental and applied, you can actually thrive, or you can actually propel what you call as the space flight biology research. So essentially, spaceflight biology research is one among the areas which are coming up in the country, which we need to build a lot of workforce to support our country's missions like Gaganyaan and further missions, including humans. So we will be looking towards Indian astronauts flying frequently, so that we need personalized medical therapies for these astronauts, as well as the countermeasures for most of the hazards that we have already noted. And one more thing, the risks and health consequences of these particular missions are also dependent upon the duration of the mission, as well as the age, the sex, and the genetics, and all these of the subjects who are traveling. So when our country is taking up the missions, irrespective of what data is available on similar studies done from other countries, we need to build our own database. Then only we can sustain our program. So many of the programs or many of the research that we may feel that it is getting repeated, it is not actually a repetition, because we have altogether a different subject which we are sending from our own soil. So that brings the difference in that particular research. Then I told about research in space laboratories. So we call them as space stations. In fact, they are one among the beautiful features of the space exploration. So you know about Sreejalekshmi and the International Space Station, the one which has been the longest inhabited by humans so far. Almost 25 years of human existence has been there. So typically, now we have wonderful news again from our Indian Space Research Organization that the country is actually setting up its own space station. We call it as Bharatiya Antariksh Station, BAS. So the mission or the completion of this particular space station is announced for 2035, and the first module will be flying by 2028 if things are working fine. So there is again lot of excitement. So two things today I'm focusing on. One is our human space programs, and the second thing is about the Bharatiya Antariksh Station. So both of them requires lot of research on microgravity, okay, under microgravity conditions. So this is a very common slide, or you all will be knowing about it, because we all, the life forms on Earth for the past 4.2 billion years have been thriving on this ubiquitous pull, what we call as gravity. And among the four fundamental forces, this gravitational force is by strength, relative strength, it is the weakest. But it operates among all objects in the universe, and thus it becomes one of the prominent forces, even though it is the weakest of the 4 categories. You can see a quote from Stephen Hawking here given. So the large scale of this universe structure, even it is actually formulated or governed by this force of gravity. So since we now know that this gravity is that particular variable which has a lot of impact on the scientific processes, if we remove that particular element, then we can understand the fundamentals of many processes that are going on, especially in living objects. So typically that brings us the since space is giving you that unique opportunity to lift out this gravity and bring up this microgravity, space becomes one of the important microgravity research platforms. So moving further, again, this is about gravity only. This is everything, most of the things listed here you already know. So we experience acceleration due to gravity of 9.8 meters per second squared. And the distance is something which governs this force of gravity. And in space, we are having what you call as microgravity. And when it comes to microgravity, we are putting actually, it is a free fall. That means the object will be experiencing weightlessness. So typically the term microgravity is used synonymously with zero gravity or weightlessness also. So we are trying to understand how this microgravity is going to impact the life processes, the biological processes. So as I mentioned, now we are in a new era for in space research. A lot of activities are going on, and futuristic programs are going on. Bharati Antariksh Station will be coming up, and then Gaganyaan flights, a host of them will be coming up. So what is that the whole country is excited about? Of course, the missions will happen and there will be a lot of science coming back. But for each individual, each researcher, each student, we get a host of opportunities to conduct the microgravity experiments, specifically this bioscience experiments. So why excited about bioscience experiments? Of course, understanding the fundamental processes in biology actually will bring a lot of benefits for subjects on Earth as well. For example, in the case of biomedical research or finding new therapeutics, this all knowledge is going to build or actually compile and give you a lot of new information, and acquired knowledge base, database can be built not only to sustain the life in space, it will also help to sustain the life on Earth. So now you may, some of you will be having the question, how will I know that such an opportunity is coming up or not? So ISRO has built a beautiful system where its website will announce what we call as AO call. It is announcement of opportunity call, where periodically you will see such opportunities for all the Indian researchers, which will be posted on its website. So please do keep a watch. So mostly you may get opportunities like pre-flight opportunities, which are preceding some major flights. Similarly, developmental flights, like for Gaganyaan, we have multiple developmental flights. So I have to caution you or warn you here that when we are trying to build biological experiments to suit the microgravity or the space laboratories, there is a huge challenge associated with that. The challenge is that on ground, I can simply add a solution to another. I simply just pour it out. But in microgravity, I have a lot of restrictions when I need to deal with the samples under microgravity conditions. So especially when it comes to biological experiments, a lot of extra care has to be taken. Because the samples are already under the stress of microgravity, and the shocks during this launch and the re-entry, all those things are going to affect the science outcome from your experiment. So what I wanted to tell is that when you are planning for a space flight experiment using biological samples, that is life on board, you have to be very clear about the hardware requirements. Okay, so the first step is when you design the experiment, you have to talk to an engineer who will be telling you about the specific requirements when the experiment has to be taken to space laboratories. So hardware is going to be the critical requirement and criticality when it comes to life science experiments on board space platforms. So in this context, I wanted to introduce two topics which are not very popular in our country at this period of time, but it is going to become very popular, and it is going to actually lead the future programs of our country, space programs of the country, especially with human space humans on board. So the two domains are space biology and bioastronautics. So this slide actually talks about what we call as space biology. So essentially, when we talk about when we are looking for a sort of a definition for space biology, broadly we can put it as the study of biological and biomedical processes in space using live specimens as experimental subjects. So when I call live specimens, how can we call a specimen as live and all? You must have different definitions for that. But as of now, several specimens, including human body, plants, food materials, and even habitat building, has been tested in various conditions of space. And when it comes to the life sciences, especially the 100-kilometer or your Van Allen belt and all, may not be the definition. So what we call is a physiological space. So physiological space is something beyond which a living object will not be able to sustain or survive on its own without any artificial supporting system. So especially around, say, 12 or 13 kilometers even, you find the starting of a physiological space. So it actually differs from species to species. And the point that I want to convey here is that when you are taking life to space laboratory, you have to understand a few points listed here as bullet points. One is the space is actually deprived of what you call as the directional stimulus. We don't have any sedimentation process happening there. The density-driven convection is not happening in space. Mechanical diffusion is prevented. The normal convections are not there. It is actually dominated by what you call as Marangoni convections. So all these processes has their own impact on the biological experiments according to the objectives that you are keeping. So what is the knowledge that currently we are having? So early years, even before our country gained independence, people have tried this life experiments in space. So initially there were balloon experiments and all. If you trace the early years of space flight using live samples, we will find that a lot of experiments have been conducted. Okay, so from those experiments, what did we learn? We learned that the living organisms are able to survive space flight. So the early years, the question was only that one, whether the living organisms will be capable of surviving space flight. Yes, later a period of time, it was confirmed that they are able to survive. And the knowledge that we gained also helped us to understand the conditions of impact of microgravity or cosmic radiation, as well as the rigors of what I said earlier, launch of re-entry, all how this is going to impact our life specimens. So those are all were the initial years and preliminary data accumulation. But currently, with that information already available, the focus has been shifted towards the changes in biology happening at cellular and molecular level, which is occurring during and after space flight. So in fact, when our country is starting to have this space flight science experiments, we are essentially contributing to this continuum of space flight science, which is already there. So we have to be more cautious and more proactive in designing experiments and contributing to this particular branch of science so that we stand out as unique researchers in this domain. So then, as, for this matter, there was deliberations which happened at ISRO headquarters related to formulation of space biology vision plan and all. And in fact, I was fortunate enough to be part of those deliberations. So essentially, the space biology vision for the country, currently we are in the first years, so say 1 to 3 years. So we have to be ready with, say, some of the experiments and facility establishments, to talking about the ground-based analog experiments, flight opportunities, designing of hardware and all, so that from small biological systems at the cellular level, like microbes, plants, animal models and all, we have to build increasingly complex systems so that we understand this developmental biology, or develop microfluidic platforms, or build animal models for drug screening. so that in the longer run, when the human explorations for our country is to be taken in a sustained form, you have to have regenerative life support systems, which will be actually a closed loop system with bioregenerative life support, which will actually support the long-term survival in space. So this is our core aim, is to have a program where the research and development will lead to technologies that will support our humans to travel to space safely and to be productive there with whatever missions that they are interested. They can do it efficiently and then come back to Earth and have a pleasant life here on Earth later. And as I mentioned here, this particular aspect is coming again and again. The critical component for any such experiment is going to be the hardware, specified or customized hardware for the experiment. And once you are ready with your some experiment, you can actually plan it in at least three different ways. Like what you know is a human in loop experiment. That is what is already ongoing in our ISS and similar space stations. Then humanoid in loop, you can use robots to control or to intervene in your experiments. That is humanoid in loop, as well as completely autonomous experiments where the experiment will be completed on its own and you can bring back the payload and analyze it for further. So, as I mentioned, along with this space biosciences emergence in the country, a host of opportunities. So this particular slide, I wanted, in case there are academicians or researchers in the early stages, of course, this 1 and 2 is the places where you can focus upon, because in the first few years, we will be locating the ground-based analogs or platforms to conduct microgravity research on Earth, laboratories, and of course the spaceflight opportunities that will be coming along with the announcement of opportunities. So you can actually identify people who are working in similar domains and set up research teams. So mostly there will be space biology proposals also coming up from ISRO side, or maybe through Respond Basket and all in the coming days. So a lot of initial years, the struggles will be more. So we need to keep us motivated along with the awareness about the challenges. And of course, the awareness about the opportunities. So that is the first phase, maybe 1 to 3 years. We are now almost, maybe we started and we are still there in the first phase only. And next is to have academic programs tailored for training the next generation biologists, especially the space biologists. So along that route, we'll be also developing labs and infrastructure, and outreach programs and public interface will be talking more about microgravity life science experiment. Essentially, that is the reason why the START program is also having one among a few of lectures which are towards this domain. So, and in the third phase, actually, we have to focus upon the spin-offs. So whatever is coming as a bonus from such experiment, they are the one which are making space programs very unique. And when you consider the human space programs of other countries, almost 3,500 plus experiments related to biomedical and biological experiments have led to what you call a spin-offs. A lot of products have come and they are all helping in various societal applications. So, and apart from that, we have to enhance partnership with external agents, internal within the country definitely, and external partnerships are also very important, because knowledge is something which need to be shared. Okay, then only it will multiply and more people will benefit. So industrial partners will come up, and a lot of startups will also get opportunities in these domains. So in the fourth segment, we will be having supporting the Bharati Antariksh Station, and from their onwards, we will be having decadal plans to be ready for the next longer duration missions. So the second domain that I was talking about is what we call as the bioastronautics. It is specifically a newer term to our Indian researchers. So there's a unique combination which we never thought about in early days, because the biologists were considered as separate entity, engineers separate, medical separate, space science altogether separate. So now every such domain has to come together and shake hands.
30 minutes over.
Okay, so I'll actually rush up a bit more. So here we have maybe 5 or 10 minutes more, and I will just try to tell you about this significance of these topics. So typically, our aim is to use the technology and engineering principles to facilitate healthy human inhabitation. So here, a very, very important challenge is to deal with the man-machine interactions under varying gravity conditions. So several challenges, health, medical risks are listed over here. Among that, microgravity is one very important thing. You can see many diseases like kidney stone, osteoporosis, muscle atrophy, or this bone mineral demineralization. All the things happen when we are having prolonged exposure of our body in microgravity. Other than that, earlier I have mentioned a few. Here also space radiation is there, isolation and sensory deprivation, this intoxication due to this artificial environment, microbial infections can happen, and unpredictable accidents, including sometimes loss of life can happen. So typically bioastronautics is actually the study and support of human or life in space. Okay, so here the aim is to protect astronauts during long flights and after long flights, to provide sufficient resources like food, water, medicine, air, and all. And along with that, the issues of gravitational biology also need to be addressed. So here, remember, the duration of the flight matters. Short duration flights may not impart this much of problems in humans, but they do have their own significant impact, but not too much. But the isolation from Earth and the distance from Earth all will actually have a say on how much problems the astronaut is facing. So I've talked about a lot of challenges. It is not to scare you, but it was rather in order to do that. Along the challenges all the time, we see multitude of opportunities as well. The first and the foremost is actually the microgravity environment, the unique environment. So experiments that are impossible to perform on Earth, you can conduct it on microgravity environments on board the space flights or satellites, or even some platforms are available. I'll talk about that. And of course, the commercial flight opportunities are of course increasing, but how affordable they are, daily research is possible. Actually, it is a tough question to answer, but I would say that we have to look for alternatives. So one of the alternatives is to use the ground-based simulation of microgravity or partial gravity conditions. How do I start? So that typically talks about from where you can start your research. Essentially, for this particular set of experiments, the ground-based facilities for simulation of micro or partial gravity conditions or even hypergravity conditions are being utilized. Why should we use them? Because they are going to define, or they are going to help you to define certain baselines, and they become valuable tools for you to prepare spaceflight experiments. And many a times they give you very important information and help you to address the gravity related issues that can be actually known prior to the real space flight. So the list of ground-based facilities are listed over here. Clinostats 2D, 3D, random positioning machine. On the middle, we are having this microgravity, this random positioning machine. And then we are having rotating wall vessel. Then you can go for diamagnetic levitation. For biological samples, it is a tough thing to subject it to such levitations, but it is done in some experiments. And there are ballistic trajectory, what we call as parabolic flights. So essentially, the parabolic flights will give you around, say, 20 seconds of microgravity, and various drop tower facilities. And this is the drop tower facility. Middle left, you find this drop tower facility as Bremen, Germany. Whereas in India, we do have a unique drop tower facility at IIT Madras. So you can actually explore their website and see. Other than that, sounding rockets are also one among the opportunity where you can conduct research. So these are again platforms where you can conduct microgravity research on your samples. So satellites offer one sort of microgravity platform, POEM platform. We'll talk about in the next slide. Then we have scientific balloons. TAFR Hyderabad is having a scientific ballooning facility. And in our IAST also, we do have a scientific ballooning facility with a range of, say, 38 kilometers. So some of the experiments are being done on scientific ballooning as well. Then we will talk about the fourth stage of PSLV, our rocket. We call it as a POEM platform. That is again another opportunity to conduct your experiment. And of course, we are having sounding rockets. You see, this is the sounding rocket launch from the Tumba Equatorial Rocket Launching Station, the one among the very prominent rocket launching stations in the globe itself. So a lot of experiments you can plan. Yeah, this is one thing I wanted. Most of you will be knowing this platform because recently in the SpaceX mission we had around say 2024 payloads which were flown on POEM-4. So especially we can, this is actually a PSLV orbital experiment module. That is what is POEM. So in space is actually authorized. I mean, it's taking care of the announcement of opportunities for hosting payloads on POEM. Please keep a watch on InSpace website. So you can plan your own experiments, the microgravity experiments using biological samples or technology demonstration in whatever way you want. You can design experiments. The capabilities of POEM platform also will be available in website. I have given a glimpse of them. So the very recent experiment, it is the CROPS, the Compact Research Module for Orbit Plant Studies. So you can see the germination of seeds. The two-leaf stage is being photographed here. It fetched a lot of attention from the scientific community as well as media within the country. So this is one example for a space biology experiment. But we will be talking about experiments that can come back to Earth as well. So what to say, retrievable payloads, we call them as. So once you conduct such experiments, why do you want to conduct? Of course, it is to optimize the ground and pre-flight experiments, which will be a good starting point for space biology experiments. So please try to use this opportunity. And research in orbit, of course, we are talking about everything indigenous, but if you see the backdrop of this particular module in the International Space Station, you see how complex it is to conduct experiments, especially biology experiments. So we need to have international collaborations. So along these lines, ISRO is currently collaborating with the NASA for conducting one Axiom-4 mission. So it will be taking our own trained astronaut, as well as some of the payloads from within the country to launch to ISS, and some science experiments are being planned. So other than that, this is two examples I wanted where my group is working on development of certain payloads for two missions. One is the Gaganyaan mission. So you can see on the left, this is my small team. So Chitra, who is moderating this program, is also found in this photograph. So we are working for developing a payload using Drosophila melanogaster, the simplest, actually very useful genetic organisms, Drosophila melanogaster. So this assembly of payload for this simple experiment is what is given in this chart. Just to tell you that it is several sub-assemblies go into the making of a special spaceflight hardware. So we do have collaborators from the agriculture universities as well. And the second payload again is with seeds. We are performing some experiments where the food crop seeds will be taken to International Space Station with the support from, again, Agricultural University of Kerala. So we will be having some 6 varieties of seeds, and our own astronauts will be trained in conducting experiments on board space. Several science objectives are also there. I'm not touching that part. So in summary, what I wanted to tell you is that lots of challenges are ahead. So we have to keep ourselves motivated. Okay, so what should motivate us? Motivations actually comes from the innovations that actually drives this particular programs, and are spin-offs from these programs, which will definitely benefit humans in space as well as on Earth. So we are looking towards disruptive innovations, disruptive technologies, and high impact scientific studies and technologies from these programs. A lot of research has contributed, has come from space and benefited on Earth. A host of them from NASA's website I have taken. And to give you an update, ISRO has transferred currently over 400 technologies to around 235 industries, out of which the major contributions came up till now from the chemicals and special materials. So once the human space programs and the Bharati Antariksha Station launches, you will find that the biomedical sector is going to actually lead with lot of benefits coming from the space programs. So we are all eagerly looking towards that. Okay, so as you all will agree, we are really proud about the Antarikshme Atmanirbhar made by ISRO, right? So are you eager to contribute and support to the space missions of the country? That is the question that I wanted to ask you. And I wish that let this start 2025 be the start of your space journey. So if you are ready to start your space journey, there are a lot of opportunities there for research, for learning, everything. I have listed a few of them. Of course, IIST, ISRO, and in Institute of Aerospace Medicine, we in India can support you in a lot of programs or areas of research. A few areas of research which are going to be very, very dominant in the coming days to support the program is also listed over there. So bionic science and engineering, bioregenerative life support system. You will find that any domain or any discipline you are in, you can actually tune you as one among the space researcher, space science researcher, space life science researcher, bioastronautics researcher, if you are willing to do so. Okay, so with that, let me actually tell you one more thing. This is a very important quote which is dear to my heart, which was by our first Chancellor, Dr. APJ Abdul Kalam. That is, small aim is a crime, have a great aim. So as Indians, as what to say, strong supporters of Indian space program, definitely I think all of you who are passionately attending this program is very, 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 very eager to contribute to this particular domain, space domain, bioastronautics, space biology, and other space sciences and technology domain. With that, I think it is time to say a word of gratitude. Of course, START 2025 organizing team for this wonderful opportunity, my institute, IIST, and our parent organization, ISRO, for all this support. Of course, my students and collaborators, they keep on working and giving more and more challenges and questions. Okay. And finally, every one of you who have attended this session, a big thanks to all of you. I hope you have grasped something from this small lecture. And in case if you are really interested to know more about our research or any queries, in case if you don't find time to answer most of them, you may please send an email to srija@iist.ac.in. And you can also visit our website at the particular website is given. So with that, let me thank you once again. And yeah, thank you all.
Thank you so much, ma'am, for the informative lecture. So there are few questions in the chat box. So the first question is, how does microgravity affect the biology of living systems?
So essentially, you know, life as the way we know it, it emerged on Earth around, say, some 4.2 billion years ago. So from that time onwards, every system in our body evolved under this particular force of gravity, where everything is actually pulled down. Okay, so when you have every system in our human body as well as in all organisms which are tailored to sort of defend this force of gravity, or maybe work along with this particular force of gravity. So the simplest example how it impacts is, once you are going to microgravity, you must be seeing some impacts of space, like puffy face, some congestion and all. So the lifting of microgravity is actually giving you clues that gravity was there when the things were evolved. Okay, so for different organisms, the impact will be in different scales depending upon various factors. So overall, there is impact even at cellular levels or molecular levels. We have evidence that gravity or microgravity is having an impact on biological systems. Okay, you can actually read more about in scientific articles are available depending upon your areas of interest. You can please read it. Okay, thank you.
Another question is by Rajeshwari Swain. So the question is, can bioengineered plants or animals provide sustainable solutions for life support systems in space habitats?
Yes, in fact, this is a very, very important question. Bioengineering or genetic engineering is one among the probabilities. But we have to remember one thing when we are artificially creating anything, be it for space or be it for Earth application, the acceptability of the particular system, especially if you are going to use the plants, means we must be using them for as a food or maybe for some other purposes. So the qualification of the system to suit the particular application has to be ensured. So there are certain guidelines which will tell you about whether acceptable ranges of changes, or because this bioengineering, no, we don't know what is the impact of it, and it has to be evaluated in long ranges. Then only it will. But this is a very promising and prominent area. Definitely, you can think about and start working in the domain, of course. Yeah.
The next question is from Niveda. She wants to know how artificially microgravity is created for astronauts training.
So typically, when we are having this, what you call as human centrifuges, okay. So for our astronaut training facility, in fact, if you have visited the Institute of Aerospace Medicine in Bangalore, or the astronaut training facility, so they are just setting it up. We use what you call as the human centrifuges. So using this human centrifuges only, the training for astronauts is generally happening. And other than that, there will be some sort of immersion tests. So different levels of tests are being done. Bed rest test. There will be a tilted, tilted bed rest test. So many tests are there. But for this gravity-related thing, we use human centrifuges. So various levels of rotations are able we provide. Okay, yeah.
Another question is that what is the time taken to recover an astronaut after they return to Earth?
Yeah, so the time taken by an astronaut to the ground conditions when he or she returns from space flight, it depends upon how much duration the particular person has survived or stayed on space, in space. And some of the astronauts like Sunita Williams, or people like, who are well versed with the space conditions, they have already a tuned body sort of thing. Okay, so it is an individual centric question. So it varies from one astronaut to another. So there is no generic answer for this particular question, but mostly for very short duration flights and all, within 2 to 3 days even, there will be complete recovery, because with the model organisms, we are observing this particular outcome when it comes to recovery from space. Yeah.
Again, another question from Rajeshwari. What are the evolutionary pressures in microgravity environments, and how do they compare to those on Earth?
Evolutionary pressure, I think what you are trying to understand is when something is under microgravity for a long time, will it get adapted to that gravity or not? So probably there is an adaptive mechanism which the organisms are expressing. Okay, so here the thing is, it is a species dependent evolution which happens, and some of them have what you call as for example, there is always a stress factor coming in whenever a living object is introduced to microgravity condition. So this stress condition can lead to so many side effects. Okay, so how how extensive this stress is being induced in the body, that will also talk about how the body will readapt and provide its own countermeasures to adapt. So again, there is no very specific question, specific answer for which is generic to all organisms. It is organism dependent, but of course there is a mechanism which is prevailing in most of them. Otherwise, they will not survive this space flight.
Okay, the next question is from Sanat Kumar Dhawan. Why are we working so much toward to develop biology research in space under microgravity conditions rather than trying it generate it on Earth? How is the microgravity generated by artificial methods be different from one in space?
That's a wonderful question, Ivan. The thing is, although we talk about analogous experiments to conduct or to mimic the microgravity conditions, till today we are not successful in having a sustained microgravity. For example, the longest one that we can get the pure microgravity on our Earth-based systems, it is not completely Earth-based system, is on this what we call as the parabolic flights. So around 20 seconds only the microgravity is existing. Whereas in the drop towers, you get about, say, 2.54 seconds and all, because generating the conditions of free fall, it is not that easy. Okay. So, and when it comes to space is the only place which offers the pure microgravity. So in quotes, I'm putting it is the pure microgravity, so obtained only in space laboratory. And that is why we are taking this much of efforts and pain to take our experiments to microgravity. And apart from that, of course, your say clinostats and random positioning machines type of things, we are able to generate up to 10 to the power minus 4G. Okay, even then it is not actually reaching that micro level. So essentially, even despite all these troubles, we are trying to take it. Okay, good question.
I think we have one last question. Is it possible for the human being to explore moon without spacesuit? Maybe in future.
Essentially, if we are born with a skin naturally protected from all the hazards of Earth, of space, we should be able to travel to space. Our space should become less harsh and maybe similar to our Earth. Either of these should happen. Then definitely we should be able to. Okay. Or else you can think about a very, skin-like material, skin mimicking material, which we can actually paste on our skin, and people will not see that any suit is worn. But even then, we are protected from space. These are all futuristic things, but wonderful, good. Yeah. Thank you. Thank you. Okay. Chitra, is it over now?
Yeah, questions are over, ma'am.
Okay.
Thank you.
Yeah. Thank you so much. Thank you.
Thank you, Ms. Chitra, for well moderating this session. Also, our sincere thanks to Professor Sreejalekshmi for presenting an insightful and informative lecture. Thank you.
Thank you so much. Thank you. Thank you. It was a pleasure. Yeah. Thank you so much. And thank you all the participants once again. Thank you.
With this, we are completing today's session, and we'll be joining back on day after tomorrow, that is 15th January at 3:30. Goodbye, and meet you all on Wednesday on the same time. Bye.
Okay. Thank you, and Jai Hind.